Cellular linguists, things that are understood about RNA translation. An RNA sequence codes for a specific amino acid. The code is 3 nucleotides to 1 amino acid, and the code is unambiguous, meaning that a specific codon only codes for 1 amino acid. But how were these things discovered? Actually, 3 scientists are at the beginning of these findings. Nirenberg and Matthaei designed the first experiment which concluded that an RNA sequence codes for a specific amino acid. To prove this, they made an mRNA containing only uracil, a homopolymer, added it to the contents of a cell cytoplasm, and finally, they included all 20 amino acids after tagging one of the amino acids with radioactivity. To put in perspective the incredible work they had to do, they had to randomly select one amino acid at a time and repeat over 20 times to circle through all possible options. This resulted in discovering that the poly-U mRNA coded for a phenylalanine protein chain. When they repeated the experiment using the rest of the homopolymers, they found that cytosine codes for proline and adenine for lysine. Interestingly, when they tried the experiment for guanine, no protein was formed at all. Good thing this wasn't the first homopolymer they tried, or it may have made them question their experiment setup, and we would have missed out on some groundbreaking discoveries. It turns out that a poly-G mRNA doesn't code for any amino acids. So Nirenberg and Matthaei have concluded that a specific RNA sequence coded for a specific amino acid, but their experimental design only worked for homopolymers. What about other RNA codes? Obviously, Nirenberg was on a quest for answers, so he paired up with Leder for this next experiment. Again, he used radioactive amino acids, one at a time, but this time they preloaded them to their respective tRNAs. Then they added them to a solution with trinucleotide sequences and ribosomes. The neat piece of equipment they used is a filter which allows everything to pass through except ribosomes. So if the ribosomes are holding on to bound trinucleotides in a tRNA charged with a radioactive amino acid, the filter will now register as radioactive. So they could tell which amino acid was attached to which codon. This must have been a very tedious process because they had to repeat the steps 20 times for each trinucleotide sequence to find the correct amino acid, and they eventually identified 61 codons. All their efforts were worth it, in my opinion, since they gave us this genetic coding dictionary found to be practically universal.